Okay, so good evening. So for today's session, we will be taking up the topics that were covered in this particular week, in week 9. And as part of that, what we will do for the day. Let me just open the PPT for today. So for week 9, first of all, we are going to do a recap of the topics that were covered, followed by the assignment questions that were presented as part of this week. And we'll go through some sample problems for the upcoming week, whose assignment you might already be very close to submitting. So for starters, just give me a second. Yeah. So first of all, we will go through the topic. So as you already know that as part of this course, we are looking at the different aspects of any living organism, starting with evolution, understanding of cells, biomolecules, the cellular processes, the physiological processes, and classification. So we are currently going through the different aspects of human physiology. And for this week, we are looking majorly at the circulatory system and the muscular system, whereas in the previous weeks, we have looked at other aspects of it. Like, for example, last week was majorly centered around digestion, little bit about the immune system as well, but majorly the topic that was focused on was digestion. So in fact, under physiology, each of these different topics are of relevance. So you start off with the circulatory system, then comes the muscular system. So as we have already discussed in the previous live interaction session as well, is that each of these systems, while they have their own significant independent function, what is important is the crosstalk or the coordination between these systems, which allows an individual to survive. And it is not that any person can survive without any of them. You need each of them to be functional and each of them to coordinate with all the other systems in order to make it possible for an organism to live. So that is what we majorly see here. And then comes the nervous system, the endocrine system, and finally the excretory system. And also that includes the digestive system, which we have already discussed in the previous week. So following that, we are now going to start the topics that were discussed in the first lecture. So what it basically looked at was the circulatory system. Now, as you understand from the nomenclature itself, it is the system which exists in the body in order to circulate or in order to transport all of the necessary components, which includes nutrients, which includes oxygen, which includes any and everything that might be required in order to allow an organism to survive. It is very similar to what you would understand as the vascular system in case of plants, where you have to. So where you have to, or where the plant actually has a system in place, like a piping system, if you think about it that way, which allows it to transport the food which is synthesized in the leaves to it throughout the plant, or which allows the plant to actually transport water from the roots to all the different parts. So similar or analogous to that system, what we have is our circulatory system, and its job is majorly, as you can see in the slide as well, is majorly transporting gases, nutrients, wastes, and also hormones. So this is actually very important, that waste which is generated throughout the body also needs to be collected, needs to be transported to the destination, which will allow its removal from an individual's body and will ensure that no person is ever left with any toxic materials within their body for an extended period of time, because that can have a lot of adverse or you can say undesirable effects, right? So what it basically composes of is a pump, which is the heart, which pumps the fluid throughout the vessel. So you can by default, or let us think about it in a way that, the general or the basal state of any system is not such that it will allow circulation to happen just by virtue of its existence. You need some sort of a push, which is provided by pumps that allow fluids to be circulated throughout the body. understand by blood. Through an open chamber. There are no blood vessels, there's no piping as such in place. Compare that to a closed system. As the name indicates, there is some sort of an enclosure through which this liquid would be, or whatever fluid we are talking about, that would be able to travel. So here the fluid yeah, I think there was some disruption. Just give me a second. Some internet issue probably. So like I was saying, a closed system would be such that fluid gets circulated throughout the body using some enclosed piping or tubing, which you understand to be as the blood vessels. Now, when we talk about the open system, then what organisms actually have it in place, and why don't they have a closed system as well, if that is the more advanced version? The reason being that they are less evolved, to put it very simply, they are less evolved. Arthropods and most mollusca, they have an open circulatory system. So if you look here, you'll be able to see that they have multiple hearts and they have openings within their heart through which blood is circulated. And where is this blood present? Within the body. If it is not enclosed, then this is actually enclosed or contained within a sort of a cavity inside the body, which is the hemocoel, which by the name you understand, hemo basically means blood. Seal is usually used for coelom, which is a cavity, a body cavity. So hemocoel is the body cavity in the system of arthropods and mollusca, where they house the entire circulation of these of the blood. So hemolymph is actually contained within the body cavity, which is known as the hemocoel. And they do not have one pumping system, they do not have one heart, they have a series of heart which is responsible for the circulation purpose. Coming to the next one, which is the closed system. So in the closed system, we have vertebrates, annelida, and a few mollusca. All of them have closed circulatory systems. So like you saw that mollusca, also some mollusks, they also have an open circulatory system. That actually depends upon the example you are citing in each case. So here what happens is that the blood circulation or the flow of blood is actually regulated by the pumping of the heart. It does not directly come in contact with the body organs. It's not an open system, as the name indicates. Here also you see multiple hearts, right? But there are vessels which are present, and these vessels are carried throughout the body of the organism so that it can perform its definite function, whether it is transporting nutrients, whether it is transporting some gas, or whatever its destined function might be. So that is what happens in a closed system. Next, we talk about the type of hearts. Then majorly you can, if you remember the discussions that we used to have about classification as well, where you saw that, or where we saw that as organisms have evolved over a period of time, apart from all of their different developments, one very direct understanding is seen in with respect to the evolution of the heart of these organisms. As it becomes more and more advanced, the circulation or the control of blood flow throughout the body is maintained by a more complex, a more you can say a more well divided sort of heart. So when we talk about the vertebrate heart, the simplest one is a two-chambered heart, which is mostly seen in fish. Where, what do you see? What is a two-chambered heart? So basically you see there is an atrium, there is a ventricle. These are the two main chambers that you need to have. What is the mechanism through which it is circulated? If you look into that, you will see that it receives this atrium. It is the recipient of all of the blood from the body. And what it does is, after collecting all of that blood, it will send that deoxygenated blood via the ventricle to the gills. So instead of lungs, what fish have, they have gills. So gills are the site where respiration or the oxygen exchange takes place and oxygen can be taken up into the body of the fish so that it can be circulated throughout the body for its use in different systems. So a ventricle sends blood to the gills in order to collect oxygen. So that is how the regulation is actually controlled. You see one atrium and you see one ventricle, and they are more than capable enough of allowing that organism to survive under the conditions that it lives in. However, with more advancement or with evolution or over the course of evolution, what is observed is a three-chambered heart. So in a three-chambered heart, the point of difference is that there are separate atria. So what it does is there's one common ventricle, but there are two atria. So it is able to segregate, or it is able to separately maintain the oxygenated and deoxygenated blood, which is a requisite, a necessity, and also an advantage for land organisms, or you can think of them as terrestrial organisms. That would include reptiles, that would include amphibia, etc. So although blood can mix in the ventricle, the mixing that will occur is quite minimal. And in fact, there might be a bulb-like structure, which you often also see in certain reptiles. So here, if you see, this is the heart where you see two atria, you see one ventricle, and from the body capillaries, blood is brought back to the atria. From there, it is pumped into the ventricle, and then it is sent to the lung, to the capillaries within the lung, in order to facilitate their oxygenation, so that this oxygenated blood can again be pumped by the heart for circulation throughout the entire body of the organism. So that is how the circulation is actually controlled. Now coming to the most you can say relevant, most advanced, most evolved, and most efficient heart that exists in the current scenario is the four-chambered heart. So in the four-chambered heart, which you see in birds, you see in mammals, what happens there is you see a complete separation of the oxygenated and deoxygenated blood. So this is necessary in order to support fast metabolism, which is found in homeotherms. So here we see so homeotherms are those which are able to regulate the body temperatures. So you see these are two atria, these are two ventricles. So as you know, deoxygenated blood from the body will be carried by the blood capillaries back into the heart via the atria. From there, it is pumped to the ventricle, which takes that blood to the lung capillaries for oxygenation. From there, it is returned back to the heart, particularly through the atrium, again to the ventricle, and the ventricle pumps it via the aorta to the entire body for oxygen supply to each and every organ, to each and every tissue that requires it for energy production, for survival, etc. So that is this complete separation, and the avoidance of any mixing between the two is a requisite in order to support the increased metabolism that we see in case of homeotherms. Now, this is a slide that showed the advantage of four-chambered heart. So we have atria, which are first contracting and forcing blood into the ventricles, following which the ventricles contract and they force blood through the arteries to the lungs and to also the rest of the body. How is it doing both things? Because you have two separate ventricles that are carrying oxygenated and deoxygenated blood respectively. It would be able to send it to either the rest of the body, or it will be able to send it to the lungs, depending on the type of blood that it is. Once the heart is relaxing, this cycle would actually, so four-chambered heart effectively would act like two separate pumps and would be able to execute its action very well. Vertebrate hearts are separated into two types of chambers. So you have the atria, you have the ventricles. Atria, or when you talk about just one, it's an atrium. It is receiving blood from the body or from the lungs, and when the atria contract, they send blood through a valve to the ventricle, which is just below it. This ventricle is then able to receive blood from the atria, and it is able to contract in order to send blood. Send blood to the body, or through the aorta, or it will send it to the lungs for oxygenation. So this is another diagrammatic representation of the heart. You would basically be able to see the four chambers. Right atrium, this is the right ventricle, then you have this left ventricle and the left atrium. So what it will do is it will, after it has collected um, all of the deoxygenated blood. So what it will do is that it will send it through the atria to the ventricles, which will then take it to the lungs via the pulmonary arteries to the right lung. And after oxygenation, it will come back via the pulmonary veins from the left lung, and will pass through each of these valves. So these valves actually, like I think we had discussed previously as well, what these valves do is they sort of act like a railway crossing barrier. If you must have seen, they execute that sort of a control what what would be allowed to pass and what not would. They're that sort of selective. I wouldn't say selective, they act as a barrier, which regulates that, the flow of blood only occurs in the correct direction and not in a reversible manner. So that's actually a very, very relevant aspect of blood circulation. Yeah. So next, a heartbeat and contraction. So when we talk about the heartbeat, what you need to remember is that the heart, while it is basically a muscle, the strongest, most efficient, the most well-controlled or muscular system or muscle within the human body, what also is necessary for that muscle to be able to or those set of muscles to be able to execute their action is their ability to or is the presence of a very well controlled, very tightly controlled system of um, electric control over the heart, which is actually making sure, like the signals that the nervous system or the electric signals that are present, which are ensuring that the nervous tissue within the heart is regulating when it would contract, when it would relax, how the beats would occur. That is extremely, extremely important. And any sort of anomaly in this, in the normal beating pattern of the heart can actually be indicative of a lot of, um, honestly, very fatal disorders. So this is something that most individuals, when particularly when they age, they keep a really tight check on it and ensure that the heartbeat is maintained as regularly, as finely as much as possible. So here you see the sinoatrial node, the SA node. What it does is it causes the atria to contract, sends the signal to the AV node to signal the ventricular contraction. So that is basically the heartbeat and how its contraction is actually occurring. Then coming to other components of the circulatory system. So what we have till now talked about is basically the heart, which is the pump that is present within the circulatory system. But what else do we need? We need blood vessels, which will carry that blood to all of the destined locations or addresses. So blood vessels, they fall into they fall into three major classes. So we have arteries, we have arterioles, which carry blood away from the heart, and we also have veins and venules, which carry blood to the heart. And the site of gaseous exchange, or not just gaseous, but also nutritional waste and gaseous exchange, is actually the capillaries, which are really thin walled, which are very intricately and quite densely connected, and they actually regulate how this exchange or these differences would actually occur. Next, coming to the arteries, they are thick walled, they are lined with smooth muscles, and they actually are quite different from the veins that we are more familiar with. So if you look at this cross section of the artery, you will see that you will see here that there are two different cross sections which are shown. Here you see an artery, here you see a vein. You will see that the muscular layer is it's much more evident, and it is much more densely situated here. You will see a capillary bed between the two. And the reason that arteries need to have such a tight or such a thick walled structure is because they are actually carrying the oxygenated blood from the lungs, from the heart to the entire body for its circulation, for its well controlled circulation. So they are carrying blood which is present at a much um, higher pressure. So they need to be able to thick walled, or they need to be thick walled in order to be able to endure that pressure, or that heavy flow of blood through that very narrow space. So that is why they need to be thick walled, and that is why they're also lined with smooth muscles. However, the arteries, they also sort of branch off, and they form these extremely thin walled sort of structures that are known as arterioles, and they can constrict to direct the blood flow and to control it, and therefore they can either increase or decrease the blood supply to the skin. So body tissues, they contain a very vast network of thin capillaries. The capillary walls are the only, they're approximately 1 cell thick, and they allow the exchange of gases, nutrients, and wastes. But capillaries are also very, like I said, they're very thin, they're very intricately connected, and therefore the space that exists within the capillary is such that only RBCs are only able to pass through it when they're arranged like in a single file, and only then are they able to go through capillaries. On the other end of the spectrum are veins, which are thin walled. Yeah, they are thin walled. If you look here, you'll be able to see that there are valves which are present. Here you see muscle contraction and how it compresses veins. So that's why when usually, somebody's taking your blood, what they'll do is they'll press on your hand to make your veins pop. And that makes it easier for them to draw blood from such individuals. And veins have fewer smooth muscle cells, but they do have valves which prevent any backflow of blood, since they carry deoxygenated or impure, or you can think of it as blood which contains waste. So they carry blood like that. That's why there's a difference that exists between the two. Now, how do valves and the skeletal muscles help these veins function? So they help because they regulate what will be the thickness, or what will be the difference in the area through, or within the cross section of the veins, and how much space would be allowed for cells to pass through, and they would effectively regulate their function. And finally, we come to venules, which are thin walled blood collectors which branch off from these veins. And there's very low pressure which is present inside the venules, and that allows capillary beds to sort of drain into them. So with this, we are done with the topics that were basically covered in the first lecture for week 9, and we will next move on to the topics covered in the second lecture, and we'll understand the components of the circulatory system in more detail, but we'll do so after a very brief break of around 2 minutes. Okay, so now we are looking at the components of the circulatory system, and among that, we will. Okay, so first we looked at the heart. We looked at how the heart is actually working. We looked at the blood vessels. We understood the difference between. Yeah, we understood the difference between arteries and veins, how they operate, what their job is, how they are structurally different, and how that is helping the way they would function. And finally, we are coming to the next component, which is the blood. So the blood is made up of majorly four components. First is the plasma. Second, the red blood cells. Third, WBCs or white blood cells. And four, the platelets. So plasma, once you remove the plasma, what you're left behind with are these remaining three. We'll look at each of them individually. First come the RBCs or the red blood cells. So RBCs or red blood cells, they lose their nucleus at the time of maturity. So they are often even called as ghosts. The reason being because they do not satisfy, if you think about it, they do not satisfy the basic definition of what a cell should be, which is it has a nucleus, it has a membrane, and so on and so forth, but they do not have, or you could call them as enucleated. That is, they do not contain a nucleus, and it actually is a part of their maturation process. So, and they are responsible for making up approximately 99% of the blood's cellular component, which is why what you see, the color of the blood that you see, it's red because they are the major component. And what is the reason that they are red in color is what we'll come to next, which is hemoglobin, a particular protein, a very interesting and very commonly used model protein that is used for studying how protein folds, how it functions, etc., which is hemoglobin. So hemoglobin is the reason that RBCs have the color red, and effectively they are providing that red color to blood as a whole as well. So it is a complex protein. It is made up of four strands. If you think about the way hemoglobin is usually described, it's written as alpha 2, beta 2. So these are two alpha chains and two beta chains, which comprise the entire protein. Apart from that, what is present, if you look at the structure here, you will see these are two chains, alpha alpha and beta beta. Apart from these, within the center of these folded proteins, what is present is a is a heme group. It's a protoporphyrin ring with it's don't need to go into much detail, but basically you think about it as a sort of a cofactor or a small chemical group that is present, which is a protoporphyrin ring with iron at the center. So these are basically iron-rich heme groups, and that is where the name of the protein also comes about. So heme groups plus the globin chains all together are providing the entire 3D structure, which is that of a hemoglobin. So each hemoglobin molecule is able or is capable of carrying four oxygen atoms, and when oxygen is present, it is turning hemoglobin this bright red color. So that is oxygenated hemoglobin basically. Now, RBCs or red blood cells or erythrocytes, what they basically do is they are carrying one of their major functions is actually to carry hemoglobin and effectively carry oxygen through the blood to each of the different organs in order to allow them to function properly. So RBCs, they have a lifespan of somewhere about 4 months. And what happens here is that the iron which you get from hemoglobin, like we talked about hemoglobin or particularly the heme groups being quite iron rich or Fe rich, what they basically do is they recycle this iron in the liver and in the spleen. And this is taken care of by the hormone erythropoietin, which is allowing erythropoiesis, which is synthesis or production of red blood cells, and that is controlled by the kidneys. It is actually stimulating the production of RBCs in the red bone marrow. So here we have a sort of schematic which represents what goes on inside the body. So under conditions of oxygen deficiency, what is happening is that it is stimulating erythropoietin production by the kidneys. So when there is a lack of oxygen, you require oxygen in the body and you require it to be circulated or transported throughout the body. You would produce erythropoietin. What that would do is that it would stimulate the production of red blood cells or RBCs in the bone marrow. And when you have more and more RBCs, what it would do is it would effectively carry more and more hemoglobin and effectively would be able to restore the levels of oxygen within the body. When there is sufficient supply of oxygen, and not just supply, but in fact, I would say circulation or abundance of oxygen in the body, under those circumstances, erythropoietin production would be stopped or would be inhibited from unnecessarily producing or unnecessarily stimulating RBCs, because too much of anything is of course not good. It can lead to adverse conditions as well, and that would complete the cycle. On the other hand, you have WBCs or white blood cells. So WBCs or white blood cells, they are basically, you can think of them as immune cells. What they do is that they defend against many different types of diseases, whatever might be the concern. They do so by recognizing foreign proteins, so that they are able to distinguish between self and non-self, which is one of the major aspects of the immune system as well, that it has this self non-self recognition, and this is majorly carried out by these WBCs. So WBCs are actually quite a diverse class, and they have many different types of proteins, each of which has very significant, very different, but very significant roles to play in the process of immune defense in an individual. So what they basically can do is that they are able to flow through or ooze through the walls of the capillaries in order to patrol the tissues and effectively then reach the lymph system. So that's what we see here. Now, this actually brings us to the immune system as well, and we are very briefly going to go through the components and the control of the immune system as well. So it actually protects an organism against any invading pathogens. It has, like, its mode of action is basically stimulating a very rapid, very sort of heightened response in an individual by an antigenic stimulus. This response is basically what you understand by an immune response. So if you have or we have two types of immunity, which are humoral and cell-mediated, or HI and CMI. So antibody complements, like we have previously talked about it also, humors are basically what is indicative of the fluid part of the body, and that is carrying the antibodies as well. So the humoral defense or humoral immunity involves the defense which is executed by antibodies or B cell receptors. And it also includes the complements and other humoral components as well. What is its job? Its job is to defend against many sort of bacterial pathogens and viruses, etc. On the other hand, CMI or cell-mediated immunity, as the name indicates, it involves some very specific types of cells which provide or which have a role to play in defending cells or providing immunity. And those are majorly of two different types, that are T cells and B cells, depending upon where they originate and what are the functions that they actually execute. So B cells are from bone marrow, T cells are from thymus. This is the site of their maturation. And these two types of cells are the major players in the cell-mediated immune response, or CMI. So they provide protection against fungi, against viruses, and many other types of intracellular facultative bacterial pathogens as well. Additionally, cancer, the primary response, or primary immune defense, which exists in any organism against cancer under normal conditions, where you would have a system in place in order to check or in order to prevent cancer from occurring in every single person you know, is actually regulated by the cell-mediated system or cell-mediated immune defense system as well. So these are the two types, and what happens during this response is what we'll look at next. So if you look at the diagram on the right as well, what you will see is that the entire immune response is a multi-step event. The first step is that there is an entry of the antigen or that foreign pathogen or that foreign substance into the cell. And after this antigen enters the APCs or antigen presenting cells, they undergo activation, whereby we have macrophages and dendritic cells which come in. And what they do, these antigen presenting cells, they engulf whatever pathogen or whatever antigen is has come in. And they, as the name indicates, they are presenting cells. They present the antigen. So they um, digest the antigen into short antigenic peptides and present them on their surface or display them on their surface via their cell surface receptors after processing. So you have APCs, which would therefore, process the antigen and present it along with MHCs, which are major histocompatibility complex antigens, MHC antigens on their cell surface. If you look here, you'll be able to see it. This is a macrophage. It took up the antigen, processed it, and presented it via MHC2. What that would do, why is it presenting on the surface? It is presenting so that it can be further recognized by T cells, which upon recognition, like specific T cells carrying TCRs or T cell receptors, cognate T cell receptors would be able to undergo activation. That would activate B cells, and these selected B cells would then undergo clonal selection, clonal expansion, and they would then differentiate in order to form plasma cells and memory cells. The plasma cells would release their B cell receptors, which are basically antibodies, while on the other hand, we will have memory cells, which will sort of act as sentinels of the immune system in order to provide a defensive, a ready to work defensive mechanism in individuals in case of a future attack or future invasion by the same pathogen. So that is the memory part of the immune system. So like we talked about the antibodies, which are part of the humoral defense, and they are simply B cell receptors or BCRs. So what they do is they are recognizing different antigenic epitopes. I think we talked about this, the epitope and the paratope. The part of the antigen that is recognized, that is binding to, recognizing, binding to, and is stimulating a response against an antigenic epitope is the antibody's paratope. What it basically consists of is that it has two heavy chains and two light chains, which are connected by several disulfide linkages. So that's a covalent linkage which is present there. It has a hinge region, which is also disulfide rich. And even within these heavy and light chains, you have variable and constant regions as well. And together, these regions, they form antigen binding sites. And these antigen binding sites, or you can also think of them as hypervariable sites. These are regions where you can have the maximum diversity, and that actually increases the repertoire of antigens which can technically be recognized. So that is what we see here. Then coming to the production of antibodies within any individual. So you have a lag, log, plateau, and a decline phase. During the lag phase, there is simply the pathogenic entry. During the log or the exponential phase, we see that after it has been identified that the antigen against this particular antibody is currently present within the system, what needs to happen is that the levels or the titer of the X antibody, or whichever one we are concerned about, that has to increase many folds. And this steady rise is indicative of the log phase or the exponential phase. Following this, we have the plateau phase. So during the plateau phase, it sort of undergoes saturation, where the equilibrium is observed between the processes of antibody synthesis and breakdown, which is catabolism or utilization. And finally, in stage 4, we see a decline where the antibody titer, it significantly falls down. So we talked about the RBCs, the WBCs, and now we come to the platelets, which are the megakaryocytes. So they are cell fragments which are very frequently used in blood clotting. They are derived from the megakaryocytes, and majorly what is observed is that they lack a nucleus. They have a significantly short lifespan, somewhere around 10 days. So platelets are not continuously circulating within an individual. Uh, Following this, we understand what is their role. They're involved in blood clotting. What they do is that they allow the release of thrombin, or they stimulate the release of thrombin, and thrombin it converts, or it catalyzes the conversion of fibrinogen into fibrin, which is sort of a sticky moiety, and that helps in binding or um, formation, and it protects the integrity of the clot. So that is what we see here. Then coming to the blood pressure, we have a systolic and a diastolic pressure. So systolic pressure is when the heart contracts. Diastolic pressure is the pressure that exists between two heartbeats. So these are the two types of blood pressure which you often see to be measured by a sphygmomanometer as well, which actually is indicative of the heart, the heart health, the individual's overall health, and the functionality of the heart. One test that is indicative of the functionality of the heart. Um, we have heard of a lot of senior members and other people around us who said that they have high blood pressure. Basically, it's called as hypertension. Why is it such a matter of concern? Is that when an individual for a prolonged period of time has such a high blood pressure, what happens is that the walls of such individuals, they keep on getting affected. And the reason for this is that under most circumstances, a high blood pressure is the result of lesser space for the blood to travel or get transported through, because it's not just a fluid, it has a large number of cells, and all these cells, they need the space, the area to actually move about. So if you see that a particular individual, they have hypertension, there is a very high chance that the walls of their blood vessels, they're sort of uh, you can say cramped maybe, or very little area is present there because of some sort of fat buildup, cholesterol buildup, etc. And that is indicative of some sort of a heart disease. Potentially, the individual suffers at that time or could suffer sometime in the near future. So those would be indicative symptoms, or you can call them as staging, where individuals are made more cautious about their health status so that they can take care and and adapt a somewhat um, healthier lifestyle in order to get better and live healthier and live longer. Uh, this is exactly what is actually mentioned here in atherosclerosis, where we have LDL, which is responsible for forming plaques that trigger inflammation. On the other hand, the immune system, it forms a very sort of a casing around the plaque that effectively ends up blocking the artery, hence leading to the increased blood pressure that we just saw. And also, these caps or these casings can rupture, which can create clots that close off an artery. So therefore, in order to prevent heart attacks, what do you need to do? One, your genetic factors matter. You can't really do much about it, but yeah, they matter. What matters more is also the environmental factors that you're subjected to. So blood LDL can be reduced by a low-fat diet that emphasizes on high fiber foods, on antioxidants, and good fats. When we talk about good fats, we mean monounsaturated fatty acids, omega-3 oils, and you would end up reducing or preferably eliminating, but yeah, relatively reducing trans fats as well. So also, the reason why physical activity is also considered to be extremely important is because it is linked to reducing the levels of LDL cholesterol in a large number of individuals. So with this, we end the topics discussed in lecture 2, which was under circulatory system. And now we will move on to the muscular system, basically with locomotion. And we'll just start that after a very brief break. Now we are starting a discussion about the muscular system. So for starters, we understand why you need the muscular system. Basically, the process of moving from any one place to another, or moving any part of your body, that is regulated by the muscular system. However, here in the first slide, what we see is locomotion. Locomotion is the movement from one location to another, from one place to another. Motion or movement can be within an individual. You can sit at a place and you can simply move your hand, you can move your head. That is a movement. But when you are getting up, moving to another place, or you are jumping to another place, you're walking to another place, when there's a shift in your geographical location or in your position, then that is locomotion. The organs which help in locomotion are locomotory or locomotive organs. Majorly, those would include the limbs, cilia, flagella, etc. What you need, as we already discussed, is the muscular system. So the muscle is one such type of tissue among 4 different types of tissues. So muscle is a specialized type of tissue and it originates from the mesoderm. Muscles contribute somewhere around 40 to 50% of an individual's body weight for a healthy adult. Basically, we mean what they do is that they are regulating the individual's movement by a number of different actions. In particular, here we see excitability, contractility, elasticity, and extensibility, which are the special characteristic features of muscle cells. So based on where they are present, muscles can be classified into A, skeletal muscles, B, visceral muscles, or C, cardiac muscles. We'll look at each of them one by one, as you might already have gone through in the lectures as well. So first are the skeletal muscles. They are associated with components which are skeletal components. Basically, they are majorly attached or majorly associated with some bone in the body, and they are regulating some sort of a motion or some sort of movement in the individual as well. If you look at them simply under a microscope, they look sort of striped. So they are simply also called as striated muscles, and these are voluntary movements that are regulated by the nervous system. So the principal action of skeletal muscles is locomotion, and it modulates the shape of an individual's body as well. The second set of muscles are the visceral muscles. Visceral muscles are in the different organs of the body. They're present in their inner walls. What do we mean by the organs? We mean, for example, the alimentary tract. So if you remember the discussion about digestion, we understood a particular motion known as peristalsis, which is responsible for moving the bolus or moving the food mass from the mouth or buccal cavity to the stomach. So the entire sort of repeated contraction, relaxation, that movement that occurs is regulated within the alimentary canal, and that is controlled by the visceral muscles. They are smooth in appearance, unlike what we discussed about skeletal muscles, where they look sort of striped. In contrast, they also are involuntary in nature, so you can't really control when and where you would, carry out peristalsis, or where you would, for example, when your respiration would take place. So all of these are carried out, or when females would undergo the process of when they would undergo cramping, etc. These are all movements that are sort of outside an individual's control, and therefore these are considered to be involuntary and are regulated by visceral muscles. Third, we have the set of cardiac muscles, which, as you understand by the name as well, these are part of the heart, where cells are assembling in sort of a branching pattern. If we talk about them in terms of their appearance, they are very, very similar to the skeletal muscles, where they have a sort of striated or striped appearance. They are involuntary as well, and therefore a nervous system is not able to directly control the activity of cardiac muscles as well. So this is actually a sort of tabulated form, which indicates the pretty much the same information. So you have skeletal muscles around bones all over the body, visceral muscles majorly in the alimentary canal and reproductive tract, and cardiac muscles somewhere around the heart. If we look at them just based on their appearance, they can appear smooth for visceral muscles, whereas they would be striped for both skeletal and cardiac muscles. But cardiac muscles also have a sort of branched appearance, so they have some sort of cross-linking which is present. Thirdly, when we talk about the nervous control, that skeletal muscles are voluntary, whereas visceral and cardiac muscles are involuntary in nature. If we talk about the purpose that they serve or the function that they control, then skeletal muscles, since they are attached to bone, they are involved in movement, whereas visceral and cardiac muscles are involved in transport and pumping respectively. Thirdly, we have next we have contraction that can occur. We will actually go through each of these different types of contraction as well. But majorly skeletal muscles are involved in short twitches and long tetranic contractions, but visceral muscles are involved in concentric, eccentric, isometric, and isotonic contractions. Finally, cardiac muscles are involved in long tetradic movements. Elasticity is the same, almost the same for all of them, but their diameters are actually quite different, and you can see the differences here as well. So here you saw the structure of a skeletal muscle, how they are long, cylindrical, with multiple nuclei. If you remember, we just discussed that RBCs are enucleated. In contrast, here you see multiple nuclei. They are organized in 3 levels. You see epimysium, endomysium, and perimysium. They have the thick connective tissue, which is the epimysium that separates muscles from each other. Collagen fibers sort of appear a little wavy, and it is connected to the perimysium. And finally, we have the endomysium as well. Endomysium is basically consisting of loose connective tissue, and all together they attribute the strength of a muscle, and endomysium also connects with the epimysium for some sort of stability. So with this, we move on to the structure of, just a second. We move on to the structure of the skeletal muscle. So muscle fibers that are lined by the plasma membrane enclosing the sarcoplasm, which is the sarcolemma. If you see here, that encloses the sarcoplasm. Muscle fibers have multiple nuclei, structure known as the syncytium, and they have a large number of glycogen granules, myoglobin. Myoglobin is also the muscle protein. And additionally, the high amount of calcium is stored in the ER. So parallel arrangement of myofilaments in sarcoplasm is one of the characteristic features of these fibers as well. So 2 types of myofilaments, which are myofibrils, are present in the muscle fibers. So we have thick and thin myofilaments made up of actin and myosin respectively. The I band, A band, we go through each of them where we talk about the entire block, the entire structure that is present, which is actually responsible for that contraction and relaxation that we are quite familiar with. So you have these the Z line, the M line, which is between the I and A band respectively, and this entire unit, this entire sarcomeric unit as we know it, is the functional unit in muscles that is allowing a muscle to contract. So like we said, actin is one of the major muscle proteins. It's polymeric in nature. We have G-actin, which is globular. We have F-actin, which is forming the multimeric fibrous structure. These F-actins, they form a sort of helix, alpha helix, by coiling around each other. And along with troponin, tropomyosin, the F-actin forms the I band. So troponin masks the active binding site for myosin on these actin filaments. So that is what we see here. Then we see the structure of the skeletal muscle also involves myosin, another very relevant, very major muscle protein, which is hexameric, and it has 2 heavy chains and it has 4 light chains. And what they basically do is that they alternate with the actin filaments in order to form the the sarcomere as we know it, they slide against each other. We will come to it actually. So if you see here, then they form dimeric filaments. The heavy chain forms dimeric filaments by forming this alpha helical structure, except at the end terminus, and the end terminal of the myosin is globular in nature. It serves sort of binding sites for other light chains as well as F actin. The N terminus has ATPase activity, and the C terminal, it meets at the H zone, whereas the N terminals, they meet at the A band margins. So troponin and tropomyosin are the two other components. I'm not going through details of that. You must have gone through it during the lecture as well. And I'm trying to wrap up this recap a little faster. So following that, we see the neuromuscular junction, which is present in skeletal muscles, where we see that you have an efferent nerve fiber of the nervous system and the muscle fiber of the muscle system. And they are connected to each other at the synapse. And it is this entire region is sort of wrapped or enclosed by Schwann cells for insulation, and the invaginated membrane-like appearance that is present in this junction is called the synaptic gutter, where the axon at the gutter where the axon neuronal ends, and these are they are effectively increasing the surface area for synaptic transmission. Then we talked about the visceral muscle. So visceral or smooth muscles, they are sort of fusiform in shape. They're majorly free in nature, which means that they form single fibers. They have much higher elasticity. Their contractility, extension abilities, etc., are also quite high. Some common proteins present are myosin, actin, troponin, tropomyosin, calponin, caldisporin, etc. For these smooth muscles, autonomic nerve fibers innervate the smooth muscles in a diffuse sort of manner, where the junctions are not connected directly, but they are able to secrete neurotransmitters at the external region, and they diffuse through the muscle fiber membrane in order to transmit the signal. Finally, these are the cardiac muscles, which, like we have previously also discussed, they're quite similar to skeletal muscles. They are striated. They are separated by dark areas, which are called as intercalated discs. They are branched and interconnected. We have cap junctions and desmosomes also, which are present in the cardiac muscle, and these are altogether called as syncytium. The heart majorly consists of two syncytium. We have or two syncytia, which are known as atrial and ventricular syncytium. Uh, Finally, in the last lecture, we have looked at the process of muscle contraction, and I'll just come back to it in like 2 minutes. Okay, so finally, we will look at the type of muscle contractions in humans. So skeletal muscles that are obviously involved in this locomotory duties are looking at the contraction of the skeletal muscle. You can basically look at them in two different classes. There are twitches and tetanic contractions, and these are the these are under the skeletal muscular contractions. On the other hand, voluntarily the movements that can be executed, they can be further distinguished as concentric, eccentric, isometric, and isotonic. So cardiac muscle contraction can be included in tetanic as well, but you can see some molecular level differences also. So here we say, if you, let me just come here, I'll zoom in. So here you see the sarcomeric unit, the two I bands at the two end, and the X zone in the middle, which is consisting of only the myosin and not the actin fibers. So these blue are indicative. If you see here, these blue are the actin fibers. These are the myosin fibers. These are myosin heads. These round globular structures, and these are the tails which are present. And when contracted, the entire sarcomere becomes much smaller in size. The H zone, it decreases, and there is a sort of sliding which occurs, and we'll actually discuss this briefly as well. So, like we said, twitches and tetanic contractions are commonly seen. So, twitches, as the name indicates, they are very short or very quick or very brief muscle contractions which occur in response to some sort of stimuli. You see muscular contraction and relaxation, but before it can actually reach the peak, the muscle starts relaxing. The second type is the tetanic contraction. In comparison to the twitch contraction, tetanic is much longer, and muscles contract, stabilize, and relax in a sequential manner. The duration of this stable condition would depend upon what is the stimulus strength. Next, we come to the concentric, eccentric, and it's isometric and isotonic contraction. So here, concentric contraction would be where you want to overcome the resistance with generated energy. The muscle would contract and shorten. Usually, it is associated with, it is the type of contraction that you see when somebody is contracting their biceps or when they're changing their joint angles. Then we see eccentric, where due to lack of energy generation and in order to overcome the external response, muscles would contract and they would lengthen. So this response is a slightly more delayed response as compared to the concentric one. Variation in the muscle length in response to an external force. So there is effectively no movement. And isotonic is where excessive force is put on the muscle initially, and later that ends up increasing the length of the muscle. This is in response to external force as well. So what happens is that you basically have a neuromuscular regulation which occurs, right? So at the end of an axon, neurotransmitters like acetylcholine are released, which diffuse from the membrane receptors in order to transmit the signals, which lead to opening of sodium channels and an influx of sodium inside the cell. This generates an action potential in the muscular fiber, travels along the entire membrane, depolarizes the muscle membrane, and that causes the release of calcium ions from the sarcoplasmic reticulum. So if you remember, we had discussed that within the ER, a very intensely high concentration of calcium is maintained inside the ER. What it effectively does is it creates a very steep gradient such that when calcium is released from the endoplasmic or the sarcoplasmic reticulum, then the increase would be very sudden, very extreme, and it would serve as a second messenger, as a signaling second messenger, and would be able to execute its downstream activities. So calcium is it is released from the sarcoplasmic reticulum, and this mediates attraction between the actin and myosin, leading to sliding alongside each other. This sliding movement results in contraction of the muscles, and the calcium pump, it actively transfers the calcium from sarcoplasm to the sarcoplasmic reticulum, and this removal results in muscle relaxation. So this is actually described by the sliding filament theory, which you might have already gone through, given by Huxley. So what exactly happens there, if I just put it very briefly, is that once calcium is released from the sarcoplasmic reticulum, it binds to tropomyosin. There is a movement of troponin, there is ATP hydrolysis, there is a sliding or there's a contraction of the actin filament by actin-myosin binding. ATP binds to myosin, releases calcium from the tropomyosin, and moves back to the sarcoplasmic reticulum, and muscle goes back to the resting condition. So there is a repolarization which is occurring in coordination with the movement or the sliding between actin and myosin, which results in shortening and lengthening of the sarcomere, and this executed in the form of contraction or or relaxation of that muscular system. So I'm not going into the details. It was very well explained in the lectures as well. So maybe you can refer to it if you need a recap for the same. So this is how normally muscle or the muscular system or muscular contraction is maintained. However, if we think about disorders, then a muscle strain is something that would occur when overstretching is resulting in sort of a tear in the muscle fiber. And this is commonly what you understand as a pulled or a torn muscle. Majorly hamstring strains are quite prevalent in individuals, in athletic individuals. Um, then in sprinters, you would see that most of them at some point of time during their athletic career have suffered from some sort of a hamstring injury. And also, if somebody has had a previous hamstring injury as well, uh, they tend to have increased risk of another injury as well. Then we come to tendinitis. So as itis indicates, it's some sort of an inflammation, in this case, inflammation of a tendon when it is overextended, overworked, and there is no rest that has been provided to that particular area. Then tendons commonly get affected in the ankles, knees, shoulders, and elbows. Then come the carpal tunnel syndrome. Majorly it's seen in individuals who have sitting jobs involving a lot of typing, gaming, or any sort of activity where the wrist is very significantly engaged. So the median nerve, it sort of becomes compressed between the carpal bones, and it could be a tumor, some wrist trauma that might have occurred, or due to repeated use of the wrist. And finally, a very severe disorder, in fact, which is muscular dystrophy. It's a genetic disorder, and that actually leads to some sort of defect in the muscle cells, defective proteins which are synthesized. There is a progressive decline in the strength of these muscles, and effectively leads to their death. And also here we see two more: myasthenia gravis, an autoimmune disorder where circulating antibodies, as you understand, it's an autoimmune disease. It is blocking the neuromuscular junctions, motors enclave, nicotinic acetylcholine receptors get blocked, and this causes muscle weakness, which you see by drooping eyelids. It expands overall to all other muscles. It leads to fatigue, and it's a very um, it's a debilitating disorder. It does not get better. It just keeps on getting worse. So that is what we see there. And finally, we come to Parkinson's, which is also a debilitating degenerative disorder of the central nervous system. Majorly, it affects muscular system, the movement. You see majorly some signs and symptoms, some motor signs and symptoms, which is involving muscle tremor, muscle rigidity, like they have uncontrollable shaking in their hands, a slowed, uh, very weak gait, and some sort of instability in the posture as well. So with this, we come to an end of the discussions for recap of this week. And and now I will just go through the assignment questions and some sample problems for week 10, and then we'll wrap up the discussion for this week. So first of all, the first question was about which of the organisms have an open circulatory system? Arthropods, Porifera, Nematoda, or Annelida. If you remember, this was a part of our initial discussion for this week's recap as well. That a large number of organisms, they have an open sort of circulatory system where blood is coming into contact with their organs, and there are no blood vessels or no sort of a system which is in place that allows its controlled transport within the body. So major organisms which are observed, or major group of organisms that are observed to have this sort of circulatory system, are arthropoda, or insects as we commonly know them. The second question is about the simplest vertebrate heart. Is A, one chambered, B, two chambered, C, three chambered, or D, four chambered? So within vertebrates, you do not so most vertebrates, they have the four-chambered heart, but yeah, if you talk about the simplest heart among them, then it is most certainly the two-chambered heart. And like you remember, there was one atria, one ventricle. There could be easy mixing between the oxygenated and deoxygenated blood. So it's not very evolved. It's not the best solution, but it is very commonly seen. So that is the two-chambered. The answer to this is the two-chambered heart. Thirdly, we have been asked how long do red blood cells live in a healthy individual? Somewhere around around 4 months. So that would give you a span of around around 120 days. So within a healthy individual, that is the approximate duration which you can expect a circulating RBC to actually persist. Let me just put this on slideshow. Then in question 4, we were asked which of the following is present in RBCs. So is it the Golgi, is it the ER ribosomes, or the cytoskeleton? So we see ER as the answer to this, because like we said, they're enucleated, they do not carry a nucleus at the time of maturation, but they do have an endoplasmic reticulum because synthesis of proteins actually has to take place. Then we come to, just a second. Yeah. Then we come to question 5, which was about erythropoietin. Where is it produced? A, kidney, B, within the pancreas, B, within the liver, C, within the pancreas, or D, within the bone marrow. So the answer to this was also, it was provided in the, if you remember, the schematic representation, which showed how exactly RBC synthesis is carried out. Then we saw there that under conditions of, sort of oxygen deprived conditions or low oxygen conditions, what happens is that more and more erythropoietin is synthesized by kidneys, which answers this question. And under those conditions, you would it would result in more and more erythropoiesis. The name of the hormone is indicative of sort of the function that it performs. So more and more RBCs would be produced, and these RBCs are then responsible for carrying more and more heme groups and more blood, and effectively more and more oxygen, which would satisfy the initial loop that was started off. And once the oxygen levels within a normal individual, once those levels have been restored, or oxygen is present in plenty, then under those conditions, erythropoiesis is no longer required, and this would inhibit further erythropoietin synthesis by the kidneys in order to execute sort of a negative feedback loop within such people, or within normal people. Let me just put it that way. So that's what we see here. Then in question 6, we have been asked that platelet cells, also known as thrombocytes, are derived from. Thrombocytes or platelets are derived from A, megakaryocytes, B, monocytes, C, myeloblasts, or D, erythrocytes. So if you remember, the blood components or blood cells would majorly be blood cells could be majorly divided into RBCs, WBCs, platelets, and plasma. So the RBCs, as we have already discussed in great detail, have different functions. Majorly, the heme group within RBC is what performs the major role of oxygen transport. But apart from that, we also have these platelets, which are thrombocytes, and they are derived from the megakaryocytes. On the other hand, the WBCs that we talked about, they are largely involved in the immune reactions, which are sort of the defensive mechanism that is kept by the cell to ensure that it has something to protect it under normal circumstances. So that is what we saw in the previous discussion as well. Then in question 7, we were asked dash protein, or which protein is responsible for the conversion of fibrinogen into sticky fibrin that helps bind the clot. So if you remember the discussion on clotting in the lecture as well, you would remember that it's an extremely crucial step, which is like in a normal individual, you need clotting to occur properly in order to prevent blood loss. If you you have watched any old documentaries, any old series, you would know that under conditions where an individual has suffered like some sort of an intense blood loss, it usually terminates with death of such individuals. It leads to multiple organ failure. So therefore, you need to have adequate supply or adequate blood supply within an individual, so as to ensure that each of their organs is well, has adequate oxygen supply, can survive. It's receiving nutrients, it is able to metabolize, and it's basically able to perform all its requisite functions. So for that to happen, you need to be able to have a mechanism in place which will ensure that in case an individual is suffering from a wound, there is immediate sealing of that wound, and that is taken care of by this entire clotting mechanism. So that is what we see here. And what we see also are that proteins that are actually responsible for this function are thrombin. So thrombin, or rather thrombin is responsible for converting fibrinogen into fibrin, which sort of acts like a glue that binds all these different components together. So that is what we see here. Then in question 8, we were asked that muscles of the alimentary canal and the reproductive tract are examples of A, skeletal, B, visceral, C, cardiac, or D, striated muscles. So I think the hand of positioning is not accurate, but yeah, the correct answer is obviously visceral muscles. They are smooth, involuntary muscles, and they are extremely important because they are performing their functions irrespective of any stimulus or from any active stimulus from our end. They are performing their functions, and they're extremely survival essential processes as well. Yeah. So after that, in question 9, we are being asked that the muscles that have sort of a striped morphology or striped morphological appearance can be controlled by the nervous system, and that possess short twitch of contractions and are present all over the body are. So if you see here, what they've provided are basically some keywords. So once you know they are striped muscles, it automatically eliminates smooth muscles. From there, you have you have skeletal and cardiac muscles as options. Apart from that, if you see the other characteristics, they are voluntary muscles. They are present all over the body, which means that they're basically in contact with bones or with the skeletal system. Then that obviously makes the correct option to be the skeletal muscles within the body, right? So cardiac muscles are present within the heart, and they are not really voluntary muscles. They do not possess short twitches or contractions that would be extremely harmful to the individual. And they are present only at one location. So that actually is satisfied by the definition of skeletal muscles. In question number 10, we are saying that the main component of the thick filament in skeletal muscles if you remember the thick and thin filament that we have just talked about. So the thick filament in the skeletal muscle is A, actin, B, tropomyosin, C, troponin, or D, myosin. So actin forms the thick filament, myosin forms the actin forms the thin filament, myosin forms the thick filament in skeletal muscle. If you remember the schematic or the diagrammatic representation of a sarcomere, which is the contractile unit in any muscle within an individual. In question 11, we are being asked that tropomyosin is a dash-like fibrous molecule. Then again, a very direct question. It's a rod-like fibrous molecule. In question 12, we have been asked that dash muscle contractions can be observed in biceps. So those are concentric muscle contractions. If you remember the different types, we had concentric, eccentric, isotonic, isometric, all these different types of muscle contractions that could be seen. In case of biceps, the type of contraction which occurs is concentric in nature, and that would answer these questions. These are directly from the lecture notes, and you can just brush them up before you go for the question. In question 13, we were asked that the dihydropyridine receptors which function in skeletal muscle is a calcium dependent voltage channel, sodium dependent, potassium dependent, or chlorine dependent. So in skeletal muscles, it's basically a calcium dependent voltage flux or voltage change that occurs. So because calcium is the mediator of the entire process, so that would actually answer this query, and option A would be the correct answer. In question 14, we are being asked that dash movement from the muscle cell generates an action potential that leads to depolarization of the inner muscle fiber. Basically, it's sodium potassium movement only that is occurring, because action potential is regulated by these two only. Calcium is the second messenger which gets released as a consequence of that action potential. So there shouldn't be much of a confusion between the two, but yeah, you need to brush up the question a little carefully before you attempt to answer such or tackle such questions. So that was in question number 14. Then in 15, we were asked that myosin is a hexameric protein which consists of two heavy chains. Okay, I'm just getting to the answer directly. It has 4 light chains and it has 2 heavy chains. If you remember the structure, it looked much like an alpha helix, and basically it had these 2 heavy chains that were wound around each other, and towards the terminus we could see these light chains, 4 light chains, that effectively form the myosin head and the myosin tail respectively. So that was the assignment, or these were the assignment questions for week 9, and I hope most of these things are clear. The lecture notes should be more than enough, but of course, if you have any question, you can get to that quite easily, not an issue at all. But apart from that, for the upcoming week, for week 10, we have a few sample questions. So what week 10 would basically look at, we will just go through some of these questions because it's the nervous system which is coming in. So first we look at, okay, so I will not be getting into details about these questions. I'll just give you an idea of the type of questions that are there and the predictive answers for those. So yeah, in question 1, we are being asked that the central nervous, the germ layer which gives rise to the central nervous system is A, mesoderm, B, endoderm, C, ectoderm, or D, the reticular layer. So the CNS or the central nervous system arises from which germ layer basically? So it is the ectoderm which gives rise to it. We'll discuss this in great detail. So today I'm just going to just go through it. In question 2, the triple membrane of the connective tissue that protects the brain from the surrounding external pressure is basically those are the meninges, the cerebral meninges, which protect the brain from external pressures. Then coming to question 3, the 2 hemispheres of the cerebrum, not cerebellum, the cerebrum, are connected with nerve fibers. Basically, the connection between the 2 cerebral hemispheres is the corpus callosum. It's the structure between these 2. It connects these 2. So if you cut the corpus callosum, an individual will no longer be able to execute any function, right? Because the two halves of the brain, or the two cerebral hemispheres, they communicate with each other. Question 4 is about the involuntary actions within the body that are controlled by A, medulla oblongata, B, the thalamus, yeah, B, the thalamus, C, the hippocampus, or D, the pons varolii. So it is the medulla oblongata which does this, the part of the hindbrain which regulates most of our involuntary movements or involuntary actions. So that's what we see there. In question number 5, we're being asked that the area deep within the brain where the pineal gland is present or where it is located is A, the thalamus, B, the epithalamus, C, the diencephalon, or D, the metathalamus. So, very direct answer to this question. It's the epithalamus, which is where the pineal gland is located within the interior regions of the brain. In question 6, we are being asked that the primary control center for reflex action is A, cerebrum. Yes, sorry. A, cerebrum. For reflex action, is it cerebrum, is it cerebellum, is it medulla oblongata, or the spinal cord? So reflexes are majorly relayed not by the central but by the peripheral nervous system. We'll come to this also in the next discussion. So it is the spinal cord which is regulating that immediate, quick, survival essential sort of a response. So that is the answer to this question. In question 7, we are being asked that the nervous system that maintains the homeostasis of internal organs and which initiates a stress response is A, the autonomic nervous system, the central nervous system, the peripheral nervous system, or the spinal nerves. The answer to this is the ANS, or the autonomic nervous system. And yeah, that answers the question. In question 8, we are being asked that the facial nerves, they originate in which part? So facial nerves, they originate in the pons varolii, the pons, and that is responsible for the facial movements as well. The question 9 shows that the electrical activity of the brain can be known from A, EEG, B, PET scans, CT scan, or D, the biopsies. Basically, an electroencephalogram or an EEG is indicative of the activity of the brain. It shows the different types of waves, and that is actually how you can decide. Like, even if an individual is in a coma, you would still be able to study their brain waves and understand, is that person really alive? Is their brain really working? They are in a comatose state, but their brain might actually be working. The rest of the body might be working. So when a person is not actually showing these minimal signs of brain activity, under those circumstances, such an individual is said to be in a vegetative state. You don't really expect, like doctors don't really expect such individuals to recover from these kind of conditions at any point of time. And under those situations, those extremely difficult situations, the family members of such individuals are asked to decide whether they would like to, whether they would opt for the individual to still be sustained or still be supported by basic life support measures, or whether they would like to let such an individual go. So that is the reason why I brought this up, is to indicate that what a simple, apart from other tests are also performed, but apart from those tests, the EEG is one of the reasons why such individuals are made to, such individuals can be assessed, their family members can be ascertained about their condition, and based on that, very major life decisions can be made. So that was question 9. And question 10, we are saying that the number of thoracic spinal nerves in humans is, I'm not going into unnecessary details. The answer to this is 12 pairs. So that's question number 10. In 11, we are being asked that the membrane that binds to neurotransmitters. Yeah. The membrane that binds to neurotransmitters and responds via depolarization or hyperpolarization is B. The postsynaptic membrane will go through the details of a synapse also pretty soon. But yeah, that's basically the answer to this question. In question 12, we are being asked that the neurocytes without dendrites and axons are found in, again, answering this question quite directly, we see D, the amacrine cell of the retina. Yeah. Then in question 13, the structures in neurons which are formed by coiling of the endoplasmic reticulum around the ribosome is dendrites, telodendria, Nissl bodies, or the myelin sheath. Then the answer to that is the myelin sheath, is the Nissl bodies. Then the leakage of ions in the neuron while traveling down an axon is prevented by neural lemma, the type of nerve conduction from the brain to the muscle in moving the arm is afferent, efferent, interneuron, or sensory neuron. It is the efferent neuron because it is moving or it is conducting from the brain to the muscle. It is executing the action via movement of the arm. So it is the efferent neuron which is executing the effect. Then in 16, the principal conductance, which is not responsible for maintaining the resting membrane potential, is A, calcium. Sodium, potassium, and chloride are responsible for maintaining the normal homeostatic or resting membrane potential within within a neuron. And that resting membrane potential is somewhere between minus 50 and minus 75 millivolts, which answers question number 17. Then in 18, we are being asked that the ion, which is responsible for increasing the positive ions and negative potential inside the axon, leading to action potential generation, is sodium. It is Na+. And the period during which the membrane cannot receive any other impulse is the refractory period, which is between the period between two consecutive action potentials is the period where a membrane is not capable of responding to other stimuli, is the refractory period. And finally, in question 20, we were asked that the small gap at the end of a neuron, which allows the signal to pass from one neuron to another, is D, the synapse, the synaptic junction, or the synapse. So with this, we come to a close for the discussion of this week. So if you remember, till now we have looked at a very detailed recap of the topics that were discussed in week 9. We looked at the previously discussed topics. We looked at some of the assignment questions. We looked at some problems for the upcoming week as well. And with this, we will wrap up the discussion for this week. I hope these lectures are helpful to you. And in case you have any doubts, please go ahead and ask your questions in the discussion forum. And you can also bring them up in the live interaction sessions in the upcoming weeks as well. We are only left with 3 more live interaction sessions for week 10, 11, and 12. And after that, you would have your exams. So if you have any previous questions from the previous week's topics as well, please bring those up if necessary. And yeah, with that, I think I'll close the session. Thank you for being patient, and I hope these are helpful for you. So good night.